Now that we have discussed the different types of movements that can be facilitated by skeletal muscles, we're going to take some time to introduce some other concepts surrounding skeletal muscles, such as classification of skeletal muscles, histology, and muscle fatigue. This presentation will serve as your introduction to the skeletal muscles. As previously discussed, muscles only shorten in their length. Muscles will never forcefully elongate in any way. As previously discussed, our skeletal muscles attach to the skeletal system and through contraction facilitate muscular movements. Certain muscles will carry out certain types of movements. So it is here we see that there are three major types of muscles, or groups that we can put our muscles in. The first major group are the prime movers, also known as our agonists. The second group are antagonists, and the third group are synergists. Each of these types of muscles will play different roles in various muscle movements. Prime movers, also known as agonists, are the first group of muscles we're going to discuss. As they sound, muscles that are classified as prime movers facilitate the major action of a muscle movement. Essentially, they contribute the majority of the tension force that's required to carry out that muscle movement. Another name, of course, is agonist, and I actually prefer to use this name for prime movers because it helps us to understand the action of this second group of muscles, the antagonists. An antagonist is any muscle that opposes the movement of a prime mover or an agonist. Let's take an example such as flexion. During flexion, the biceps brachii muscle is going to contract and pull the hand up to the shoulder. That decreases the angle between the ulna and the humerus. So in terms of flexion of the lower arm, the biceps brachii is going to be our prime mover. However, there's another muscle on the back side of the arm, known as the triceps brachii, that serves as the antagonist to the prime mover. The triceps brachii will facilitate extension of the lower arm, and therefore it opposes the movement of the biceps brachii. When we consider a unique muscle movement such as flexion, the biceps brachii muscle is the agonist, and the triceps brachii is the antagonist. Conversely, if we were to consider extension, the triceps brachii becomes the agonist and the biceps brachii becomes the antagonist. Now you might be wondering, why on earth would we have an antagonist muscle opposing the movement of our prime mover? Why would we want to oppose the movement that's being facilitated by one of our other muscles? And the answer to that question is control. Antagonists help us to facilitate smooth muscle movements. By opposing the agonist, we can much better control the actual movement of our muscles. Synergists are our last type of muscle. Synergists, also known as fixators or stabilizers, do exactly as they sound. Synergists will work synergistically with the prime movers to either stabilize the joint while the prime mover is doing the majority of the muscle movement, or perhaps synergists might aid the prime mover and do a smaller amount of work in the muscle contraction. Depending on the muscle action or movement that we are looking at, different types of muscles can gain these three classifications. Now that we have distinguished between the three different types of muscles, we're now going to switch gears and start looking at the structural organization of muscle fibers. A muscle fiber is just a fancy name for a muscle cell. Skeletal muscle cells in general are very long and cylindrical in their shape. And it is because of this shape that they resemble a fiber, and that's why we call them muscle fibers. Muscular tissue as a whole is one of our four principal types of tissues. And what made muscular tissue special is its ability to contract. This specialized function of muscle comes from the contractile organelle inside of our muscle fibers. It is the myofibril that will facilitate contraction inside of a muscle fiber. This is the contractile organelle that makes skeletal muscles so unique. Myofibrils are cylindrical in their shape, and they're made up of repeating units known as sarcomeres. Sarcomeres are the contractile unit of the myofibril, which is the contractile organelle. And we'll also see that we can break down the sarcomere into the individual myofilaments, or muscular protein filaments that facilitate the contraction. Two of the major myofilaments that I will mention are myosin and actin. Inside of the sarcomere, these two proteins will overlap with one another. And when muscles contract, the filaments will appear to slide past one another. This is known as the sliding filament theory. It is myosin and actin which will be the most fundamental players in facilitating muscle contraction. So let's take a look at a muscle fiber and break down this structural organization. Of course, there has to be a cell membrane. With muscle fibers specifically, we call the plasma membrane the sarcolemma. Inside of the long cylindrically shaped muscle fibers, we'll find many of these myofibrils, which are themselves cylindrical in their shape. As we zoom into the muscle fiber, you can see that this is a myofibril, and there are individual sarcomeres that are making up this structure. You can see a sarcomere is stretching from here to here, and we also have a part of the sarcomere shown over here as well. There are quite a few things that can be found inside of muscle fibers, but it is the sarcomere that is of greatest importance to us in this chapter. So we're going to take some time to go over the structure of the sarcomere and the different myofilaments and their arrangement. So first and foremost, what we can see is that myosin and actin are the major myofilaments that are going to be making up the sarcomere. Myosin composes what is known as the thick filament, and actin composes what's known as the thin filament. And you can see that based on this picture, the myosin is shown in red, the actin is shown in blue, and you can clearly see why this is called the thick filament and this is called the thin filament. A myosin molecule is shaped like two golf clubs that have their handles wrapped around each other, and these little myosin heads will protrude off the sides. As muscle contraction is carried out, these myosin heads will bind to the actin molecule, and that will lead to the shortening of our sarcomere. If we shorten all of our sarcomeres, we get an overall decrease in our muscle length, which is what is truly going on during muscle contraction. There is a third myofilament known as titin. This is the elastic filament, and this keeps myosin anchored to what's known as the Z disc. I'll explain this in just a second. So up in this picture, you can see the thick filament here in red, and the thin filament over here in blue. As these proteins are overlapping with one another, they're able to contact each other and slide past one another. As these actin molecules slide closer to each other in the middle, that's going to produce the shortening of our muscle length. Now right in the center of the sarcomere, we have the M line. I would like to think that the M in M line stands for middle, or perhaps myosin. So this is going to be the middle portion of the sarcomere where the myosin is attached. Myosin will also be attached to the titin filaments, and this will make sure that myosin doesn't lose its place. We'll also come to see that the Z disc is found on either end of the sarcomere, and the Z disc is named based on its zigzag-shaped structure. The actin molecules are what are going to be bound to the Z disc on either end, and each sarcomere spans the distance between two Z discs. And you'll see that another sarcomere will start over here, and then we'll have an additional sarcomere on the other side, and that will make up what we know as our myofibril. So we have myosin and we have actin, and those filaments are going to slide past one another to lead to a shortening of our muscle. We have the M line that sits right in the center of the sarcomere, and the two Z discs that contain the sarcomere. So these are the major structures of the sarcomere. However, there's one other concept I want to share with you regarding the myofibril, and they are the A band and the I band. The A band stretches from one end of the myosin molecules to the other end of the myosin molecule. Essentially, the A band is the length of the myosin molecules, while the I band stretches from the end of one myosin molecule in one sarcomere to the end of a different myosin molecule in the neighboring sarcomere. So the I band naturally doesn't have any myosin contained within it. It is the A band and the I band that give us what we know as striations. You may remember briefly hearing about striations when we talked about muscular tissue when we were covering the four principal types of tissues. Striations are stripes, light and dark patterns that can be seen on muscle cells. And it is the I band that makes up the light band and the A band that makes up the dark band. So it is because we have lots of sarcomeres that are all sitting in line, all going in the same orientation, that we will have this dark and light pattern that we know as striations. The structural organization of muscle does not stop when we get to the muscle cell. Essentially, our muscle cell is built up by taking myofilaments, and that forms a sarcomere. We'll attach many sarcomeres at their ends, and that will form the myofibril, our contractile organelle of muscle fibers, and then we'll take many different myofibrils and pack them inside of our muscle cells. However, outside of the cell, we will see that our muscular structural organization will continue. Specifically, there are connective tissue layers that will wrap around and separate muscle fibers. And there's a very unique arrangement to the connective tissue that can be found in our muscles. The three different layers of connective tissue that I will mention are the endomysium, the perimysium, and the epimysium. Those are our three connective tissue layers that will continue the structural organization of our muscles. So the endomysium is a special layer of connective tissue that wraps around every single individual muscle fiber. Every single muscle fiber will have its own individual layer of endomysium wrapping around it. And as muscles are made up of many different muscle fibers, we'll take lots of those individual muscle fibers that are wrapped in endomysium and stick them all together. Once we have a group of endomysium-wrapped muscle fibers, we're going to wrap that in a second layer of connective tissue known as perimysium. And it is once we have our perimysium wrapping around a bundle of muscle fibers that we're going to call that bundle a fascicle. So specifically, a muscle fascicle contains a layer of perimysium wrapped around muscle fibers that are all individually wrapped up in the endomysium. And then, once we have a fascicle wrapped up in perimysium, we're going to take all of our muscle fascicles and wrap one more layer of connective tissue around the entire muscle, known as the epimysium. This picture will demonstrate these three connective tissue layers. You can see that this is depicting an individual muscle fiber, and we will wrap each and every individual muscle fiber in its own layer of endomysium. We'll take a bunch of these wrapped muscle fibers, bundle them up, and then wrap them in an additional layer of connective tissue known as the perimysium. And this is called a fascicle. We will then take all of our fascicles and wrap them up in one more layer of connective tissue known as the epimysium. And it is the epimysium that connects to the tendons that attach our muscles to bones. So in the last video, I described the difference between a tendon and a ligament. Tendons are specifically attaching muscles to bones, while ligaments are attaching bones to bones. So we don't talk as much about ligaments, we want to be focused on tendons here. Tendons are typically cord-like in their shape. They typically assume a very long and cylindrical shape. However, some tendons tend to be more flat and sheet-like. Those are known as aponeuroses. An aponeurosis is simply a flat sheet-like tendon. Aponeuroses will also attach muscles to bone, but these will typically be attached to muscles that are flat themselves. Large, bulky, and round muscles tend to have tendons, but flatter, smaller muscles will often have aponeuroses attaching them to bones instead. We'll also have one other type of tissue inside of our muscles known as fascia, and this is just an additional type of connective tissue that aids in bundling our muscle fibers together. This picture just shows us another depiction of the structural organization of muscle. But I like this picture because it goes into a little bit more detail. So first and foremost, we have our tendon that attaches to the epimysium that wraps around our entire muscle. Our muscle is just a bunch of fascicles that have all been wrapped up together in that epimysium. Each individual fascicle, which you can see here, is going to be wrapped up in perimysium, and then each individual muscle fiber wrapped up in its own endomysium will be found inside of the fascicle. This here is a muscle fiber, and inside of this muscle fiber, we can see that there are lots and lots of myofibrils that are stuffed inside of the sarcolemma. So even though it is skeletal muscle that we are primarily going to be focusing on in Anatomy and Physiology 1, I do want to take a quick second to show you some slides of all three types of muscles. Now the skeletal muscle slides is a picture that we've definitely seen before. We covered this when we were going over tissues. Skeletal muscles are very long and cylindrical in their shape, and so what we're looking at here is a bunch of horizontally arranged muscle fibers. This here is a muscle fiber, and this here is a muscle fiber, and so forth. Each and every one of these is a muscle fiber coursing from right to left. Skeletal muscle is our only type of voluntary muscle that we have. We'll come to see that cardiac muscle and smooth muscle are two types of involuntary muscle. We don't get to consciously control those muscles like we do our skeletal muscles. This slide nicely shows us the striations that are imposed by the A band and the I band. You can see this is a dark band, and we have a light band right next to it, and then we have another dark band, and a light band, and a dark band, and so forth. Those are indicating the A band and the I band, where we have myosin and actin very nice and organized inside of our myofibrils. We will also come to see that skeletal muscle fibers are quite long and quite large. These cells will have many different nuclei, and they're typically found peripherally located on the sides of our cells. You can see that there's a peripherally located nucleus here, and here, and here. And all of these nuclei will be found on the periphery of our cells. Now, you might be a little confused over here. It may look like these nuclei are actually in the middle of the cell. However, that is not the case. These nuclei either belong to this muscle fiber and are in front of the section, or perhaps these were the nuclei of the muscle fiber that got sliced off so that we could see this picture. So I want you to be very comfortable at identifying skeletal muscle under the microscope and differentiating this type from our other two. Smooth muscle is the next type of muscle that we're going to mention. Smooth muscle is an involuntary type of muscle because we don't consciously control this. Smooth muscle can be found on the urinary bladder, along the walls of our organs of digestion, and also in the uterus. Smooth muscle is the one type of muscle that is non-striated, and that's actually why we call it smooth muscle. Smooth muscle cells tend to have a very plump center and taper on either end. We often describe smooth muscles as being cigar-like in their shapes. So this is a very nice smooth muscle cell that's skinnier towards its end, but it's very plump in the center. We'll have a singular nucleus that can be found in here, and we will still have myofibrils. Smooth muscle not having striations doesn't mean that we don't have myofibrils and sarcomeres inside of this type of muscle tissue. In skeletal muscle, we saw that the myofibrils were all regularly arranged together, and that's what gives us the appearance of striations. With smooth muscles, we'll still have myofibrils, but they'll be oriented less regularly. So we end up not seeing any striations with this type of cell. But we want to remember that as a type of muscle tissue, this will still have the contractile organelle that makes muscle tissue unique. Our third and final type of muscle is cardiac muscle. Cardiac muscle is also a type of striated muscle, and we can clearly see that from this picture. Cardiac muscle is quite different from skeletal muscle, however, in its cellular shape and also in the fact that it is involuntary. We can't consciously control how quickly our heart beats. Cardiac muscle tends to have a branched cellular arrangement, and you can see these very oddly shaped cells that are shown on this slide. These cells are not long and cylindrical like skeletal muscle fibers are. The other thing that we will see with cardiac muscle cells that we want to keep in mind are the intercalated discs, and those are what can be seen here, here, and here. You'll see many more of these intercalated discs scattered throughout this picture. Intercalated discs are only found on cardiac muscle, so if you see them, you know for a fact that you're looking at cardiac muscle tissue. Intercalated discs contain two very specialized junctions, specifically gap junctions and desmosomes. As cardiac muscle is contracting just about 1 time every second for our entire life, intercalated discs need to have desmosomes to keep all of these cells attached to each other in a structurally sound network. We'll also find that gap junctions inside of the intercalated discs will facilitate quick diffusion of calcium and other substances inside of our cells to allow for cardiac muscle to function as a unit. And that means that all of the cardiac muscle cells in one part of the heart will contract at the same time. The last concept that we're going to discuss in this presentation is muscle fatigue. We still don't have the full story about muscle fatigue, but what we do know about muscle fatigue is that it can be caused by lack of stimuli such as calcium, nervous impulse, or ATP. In order for us to understand muscle fatigue, we need to understand a little bit about metabolism and where ATP comes from. So we want to keep in mind that all cells in our body use ATP as their primary source of energy. It is adenosine triphosphate that powers all of our cells, especially our muscle fibers. As you will cover in more detail in your lecture class, the process of muscle contraction is referred to as cross-bridge cycling. There are several stages of cross-bridge cycling, namely ATP hydrolysis, cross-bridge formation, the power stroke, and the detachment of myosin from actin. By hydrolyzing ATP, myosin ends up binding to actin, and we can have the sliding of filaments and the shortening of our muscle length. This is only allowed when ATP is present, but we'll also come to find that ATP is also necessary in order to facilitate the detachment of myosin from actin. So in order to facilitate muscle contraction, ATP is a must. So where do our muscle cells get ATP from? The sources of ATP that our muscle cells will be able to tap into are shown here. Each of these sources is listed top down in the order that they are accessed by our muscle cells. We'll first make sure that we used up all of the stored ATP that can be found in our muscles. There will be a very small amount of stored ATP. This is not that much ATP, however, and it only facilitates muscle contraction for about 6 seconds. It is creatine phosphate, anaerobic glycolysis, and aerobic respiration that are the major reactions that we're going to use to generate more ATP when our muscles run out. Creatine phosphate is actually a rather simple molecule that is simply a creatine molecule bound to a phosphate group. In order to synthesize the molecule creatine phosphate, we will use the enzyme creatine kinase to facilitate this reaction. First and foremost, creatine kinase is going to take creatine and ATP, and it's going to lyse off the last phosphate group from ATP, leaving us with ADP. It will then take that phosphate group and stick it onto creatine, and that gives us creatine phosphate. Creatine phosphate is a storage molecule. It stores phosphate for later use. As we start to break down our stored ATP, our ADP concentrations are going to go up. And as ADP concentrations go up, creatine phosphate is then going to grab onto that ADP and break off that phosphate from creatine phosphate and put it back on to ADP. And so therefore, we've transferred the phosphate from ATP back into this creatine phosphate and then back into ATP so that we can use this ATP for muscle contraction. This is the first mechanism that we will utilize after all of our stored ATP has been used up. Creatine kinase will facilitate both of these reactions. Anaerobic glycolysis is another concept that you will likely have covered in Chapter 25 in your lecture course. The major takeaway from glycolysis is that we take glucose and turn it into 2 molecules of pyruvic acid. And in the process of this conversion, this reaction requires 2 molecules of ATP to facilitate it. However, as we continue on to convert all of these different molecules out to pyruvate, that's going to give us 4 more molecules of ATP for a net gain of 2 ATP every time we break down a molecule of glucose. So this is the second method by which we can access more ATP in case our creatine phosphate stores run out as well. Aerobic respiration, also known as cellular respiration, is the third and final mechanism that our body has to generate more ATP. And aerobic respiration is the major idea of Chapter 25. We have 4 major reactions that make up aerobic respiration: glycolysis, the formation of acetyl coenzyme A, the Krebs cycle, and the electron transport chain. And 1 molecule of glucose running through these 4 reactions will give us a total of 32 ATP. So it's aerobic respiration that allows us to facilitate exercise and muscle contraction for very long periods of time. 2 molecules of ATP were coming out of glycolysis, a couple more molecules are going to come out of the Krebs cycle, but through the electron transport chain we'll end up forming a grand total of 32 ATP molecules. So what we can see from this slide is that the ATP that's stored in our muscle is always going to be used first, and this gives us our first 6 seconds of exercise. Oftentimes, we're going to be doing more than just 6 seconds of exercise, so we'll need to tap into our creatine phosphate stores, which will facilitate another 10 seconds of exercise. Now, if we're going to do anything more than walk down the stairs, we're going to need more than 16 seconds of energy. So therefore, that's where we're going to start tapping into anaerobic glycolysis. Anaerobic glycolysis will give us enough ATP to facilitate exercise for the first minute. The key is anaerobic glycolysis doesn't use oxygen, so if oxygen isn't present, we're going to start to fatigue after this first minute. It is only until we start to tap into cellular respiration or aerobic respiration, which requires oxygen, that we'll be able to exercise for much longer periods of time. So the lab experiment that is typically done in the lab compares muscle contraction under aerobic circumstances with muscle contraction under anaerobic conditions. Typically, we do this experiment with the blood pressure cuff. The first muscle contraction is completely unrestrained. We will not add the blood pressure cuff. And what we know from this is that oxygen is able to get transported to the muscles, and we will have a much greater ability to contract our muscles longer. However, with the blood pressure cuff on and inflated, this is going to close off the vessels that carry oxygen out to those muscles. This simulates anaerobic conditions, and therefore with the blood pressure cuff on, we will generally find that our muscle contraction is not able to persist as long as it was able to without the blood pressure cuff.